Hi everyone, my name is Alaya and I'm going to talk today about protein synthesis. Before I'm talking about protein synthesis and why it's important to make a protein in our cells, let's talk first about DNA and RNA. DNA is the abbreviation of deoxyribonucleic acid and RNA is ribonucleic acid. As you can see, DNA is a double strand while RNA is a single strand. DNA and RNA, they have in their structures consist of the same thing, which is phosphate groups, sugars, and the nitrogen bases. And the phosphate and the sugar is alternate in both DNA and RNA to make the backbone of the DNA and RNA. DNA has the DNA has a double strand and RNA is a single strand, and they have a little difference in their nitrogen bases. Not only the shape, the nitrogen basis is different. DNA has a cytosine, which is a simple C guanine, simple G adenine, A and thymine T. RNA has the same nitrogen bases sharing with the DNA, same as cytosine, guanine, adenine, but instead of thymine, we alternate with uracil. So as we now talked about DNA and RNA, and as we know, our DNA is the blueprint of our living organism, and a protein is important, the cell to make, because it's protein is important for the cell structure. So here is my post and here is my nucleus, where the DNA is here, and that's my cytoplasm. So and the nucleus is double strand DNA, and as we notice here is the DNA is unwound and open, to make the preparation for protein synthesis stage. So the first stage is the transcription. So the transcription happens when we want to copy a gene of the DNA, and this happens with the help with RNA polymerase to create the messenger RNA. So when we create the messenger RNA and we complement the nitrogen bases of the DNA to the RNA, this is a procedure called transcriptions. So messenger RNA, after is completed, the DNA will close after the messenger RNA will leave, and our messenger RNA leaves now the nucleus to go to the cytoplasm, specifically to the ribosomes. How the messenger RNA leaves? It leaves through the nuclear pores because it's a single strand and it's simpler than the DNA. So messenger RNA now is in the ribosomes. And every messenger RNA can be read by a triplet, triplet basis. So AUG, AAG, UUU, and it's keep going like this, but always read by three triplets, by triplet code, okay, which is it's a three bases of the letters. So this is the messenger RNA. Now, where in the ribosomes, what's going to happen is the translation process. The translation process is like the messenger RNA now will be translated to be amino acids. So the amino acid were brought by transfer RNA, which the transfer RNA will bring to the ribosomes the amino acids, and transfer RNA will complement the messenger RNA code by anticodons. So here we have AUG, which AUG is the start code for the translation. Without AUG, we cannot start the translation. And we see here is a complement, which is anticodon, UAC. And now the amino acids, like the one transfer RNA, has now, if the codon and anticodon matches, the transfer RNA will drop the amino acid. Each of one of these symbols, AUG, AEG, has a name of amino acid. We're not expecting to know these amino acids by our heart. So there are 20 amino acids in our bodies. So we have a list here. As you can see, AUG. So AUG, which always going to be the start codon, which is methionine, okay, methionine. Okay, so methionine. Then now, so the transfer RNA after complement the code will leave will drop off the amino acid, and transfer RNA will leave. So drop the amino acid and will leave the ribosomes. Then we'll go then another transfer RNA will come. So now is now AAG, which will complement to anticodon. Okay, so if it complement again, now AAG amino acid is lysine, so now the transfer RNA will drop the amino acid and will leave, and GGC, until we reach the stop codon, which is here. It's UAA. We have 3 stop codons: UAA, UGA, and UAG. So when we reach stop codons, we don't have a transfer RNA code because this is a blocker. That means we stop now a translation of the process. So as we said, protein synthesis is 2 stages: transcriptions, which is copy of the DNA gene through RNA polymerase to messenger RNA, and translation. It's a translation happens in the ribosome, which is translation of the messenger RNA codes to amino acids. So this is what about protein synthesis. We need to talk about a mutation. A mutation happens for 2 reasons. Mistakes happen when we made our DNA, and the other one is environmental. Environmental factors like a UV rays, like radiations. We have two types of mutations. First one is a point mutation, which happen only in single point, like it's a single base. So like here, like example, like here in the AAG. So imagine AAG, which is lysine, we change it to AAU. Okay, we're still gonna get amino acid because there is an amino acid for AAU, which is called asparagine. Okay, so we're still gonna get it, and we're gonna complete, and we're gonna have a protein, but this protein is a mutant protein. The other things is called a frame shift mutation. Frame shift mutation, it is adding or deleting one of the nitrogen base, and this happens from here, which will change the whole change of a protein. So instead, like let's say TACTT, we will remove this A, and if you remove this A, instead this AAA will happen UUU. We delete this A, so now we have to read it from AAC, so now it's UUG. This is totally different code, and this will affect the whole process of amino acids, and this will always alter the gene expressions. The mutation can be expressed in different ways. There's a silence mutation. This will not affect the amino acid or the gene or the protein. And the other one is missense mutation, which this mutation usually will create a protein, but this protein will work like less effective or will not gonna work at all. So this is what I wanted only to talk about protein synthesis. I'm hoping everybody enjoyed the video and made it easy to everyone to understand the concept of protein synthesis. Thank you.